Yeah. Live streaming, sir. Okay. Very good morning to all of you. Firstly, it's privilege for this today's state level online. One of my colleague, Dr. Amit Nerkar sir, as a resource person, and the topic medicinal chemistry and its role. As a NAC coordinator, I firstly welcome Dr. Amit Nerkar sir as a resource person for today's online webinar, and all the participants who are joining, welcome to this webinar.
Thank you, sir.
The main motto behind this webinar, as the students academics, most of the times we require for the competitive examination, there are the certain issues are there which is not being a part in our syllabus. So to boosting our student, it is a responsibility of the IQAC as well as the NAC1 to boost the various issues for the student purpose only. And for this, I am really thankful to Dr. Amit Nerkar sir. On a short notice, he accepted our invitation and given a talk on the topic of medicinal chemistry. Now I requested Dr. Ankush Wakode sir, Head Department of Chemistry, Arts and Science College, Pulga, and Dr. Ashok Davande sir, IQAC Coordinator and Head Department of Microbiology, Arts and Science College, Pulga, Dr. Manisha Ajmire Madam, Assistant Professor, Department of Chemistry, Arts and Science College. Now, firstly, I requested Dr. Ashok Davande sir, IQAC Coordinator, to give the IQAC remark.
Thank you, sir. Good morning, one and all. I am Ashok Davande, IQAC Coordinator, Arts and Science College, Pulgaon. On the behalf of IQAC, I welcome today's resource person, Dr. Amit Nerkar sir, Professor and Head from Siddhant College, Pune, Maharashtra. Warm word of welcome to our Principal, Dr. Vilas Hadge sir, convener of this program, Dr. Ankush Wakode sir, Dr. Gulani Madam, all my teaching staff, all my dear students. In my opinion, conducting such kind of program is very important. Today, so we will talk on this medicinal chemistry and its role. This kind of program is really helpful for the student to grasp the new concept and the new technique. What are the research trend in the chemistry? Today we have resource person. He will share his invaluable insight, expertise, and experience with all our attendees. And I'm sure this program will be helpful to all our students. I'm also thankful to sir for spending your valuable time with our student. Thank you. Over to sir.
Thank you, sir. Now Dr. Ankush Wakode sir, Head Department of Chemistry, gives his remark.
Very good morning, one and all. I am Dr. Ankush Wakode, Assistant Professor and Head Department of Chemistry, Art and Science College, Pulgaon. From the last couple of months, Department of Chemistry and Microbiology, Art and Science College, Pulgaon, we are looking for organize webinar on the topic medicinal chemistry and its role. And finally, today we have expert with us, respected Dr. Amit Nerkar sir, Professor and Research Head, Siddhant College Pharmacy, Pune, with us and to deliver this talk on this topic. So this webinar specially organized for BSc and MS students, and the objectives of these seminars are to focus on research opportunity for the chemistry students, to fulfill the gap between rural and urban category, to aware the students about recent research in medicinal chemistry, to fulfill the students' demands and curiosity regarding their career in chemistry after UG and PG, and to orient the student to focus on study and their goal, etc. And I would like to say that organic chemistry students can play a very important role in the drug synthesis, and if they will get a proper guidance from the expert. And so today we have the expert with us, and definitely this is our main objective behind it. And to cover all these things, today we organize this kind of webinar, and it is under the student development program from the Art and Science College, Pulgaon. So I am sure about this webinar will be fruitful and our student will get benefited from this. So thank you. Over to you, Bhanganas. Over to you.
Thank you, sir. Now, to start with our program, it's my responsibility to introduce today's speaker, Dr. Amit Nerkar. Yes. Just a minute. Yes. Justly in briefly, I introduce today's speaker, a renowned person, Dr. Amit Nerkar. Sir has 18 years of experience in teaching and research. Sir obtained a Bachelor of Pharmacy from Vidya Bharati College, Amravati, affiliated to Sant Gadge Baba Amravati University, Amravati. Also M.Pharm in Pharmaceutical Chemistry from the College of Pharmaceutical Sciences, Government Aided, Behrampur, Orissa, affiliated to Biju Patnaik University of Technology. Sir obtained his doctorate, PhD in Pharmaceutical Sciences, NIMS University, Mumbai. During his PhD, he was awarded a Senior Research Fellow of Indian Council of Medical Research, Ministry of Health and Family Welfare, New Delhi. Sir has 52 presentations in a conference of national and international levels and 66 publications in SCI Index journals with SCG INGO Impact Factor. He is also a member of the editorial board of many SCI Index journal. Sir is an well proficient with organic chemistry, medicinal chemistry, pharmacology. Sir has a research funding grant from the central funding agencies of India. Yeah. Also passed his online certification in clinical pharmacology from the Harvard Medical School, USA, and a National Institute of Health, Department of Health and Human Services, Office of Clinical Research, Government of USA. He is also a GPAT qualified with All India rank 635 in 2019. Sir presently working as the professor UG and a research head at Siddhant College of Pharmacy, Pune, Maharashtra. He is also working as an editorial-in-chief for his journal Current Trends in Pharmacy and Pharmaceutical Chemistry. And presently, sir is a PhD supervisor in pharmacy. On this note, now I requested Dr. Amit Nerkar sir to take a charge and talk on the medicinal chemistry and its role.
Okay.
Over to Nerkar sir.
Okay, sir. That was. Am I audible? Am I audible? Hello.
Yes, sir.
Yes, sir.
Yes, sir.
Yeah. That was really all beautiful introduction, and I am really obliged with Arts and Science College Pulgaon, and especially Banginwar sir for calling me here. Well, I will give all the acknowledgements at the end of the seminar. But since we are running short of time, we will start directly with today's webinar, and which is entitled as Medicinal Chemistry and its Role. So everybody is very well acquainted with the term chemistry, and everybody is acquainted with the term. Some of you may be acquainted with the term, some of you may not be acquainted with the term, but everybody knows organic chemistry, and the applied organic chemistry to medicines is medicinal chemistry. So contents for today's webinar include role of medicinal chemistry in therapeutics, which is revisited, then medicinal chemistry as applied science, and then natural products and phytochemistry as medicinal chemistry approach. And if possible, we will be looking at computer-aided drug design, which is called as CAD. Now, since the webinar timing is from 11:30 to 1:30 p.m. Mostly in 1.5 hours, I may be able to cover the 3 topics, which is up to natural products and phytochemistry and medicinal chemistry approach. Maybe CAD we will see someday later, or maybe in some another seminar, most probably at your college. It will be great. Okay. So role of medicinal chemistry in therapeutics, which is revisited here, and this is presented by me, who is working as professor and UG research at Siddhant College of Pharmacy, founder and director for Etios Foundation, founder and director for Caroline Therapeutics, editor-in-chief for Current Trends in Pharmacy, and founder and director for Afsar International Publisher, which is a publisher for scientific books. Okay, so contents for today's this part of the section is introduction to receptor, pathophysiological role, structure activity relationship, drug selection, role of drug in therapeutics, role of combination drug therapy, drug discovery for new targets, and then we come to the conclusion and summary. These are the contents for this part of the this part of the chapter, which is medicinal chemistry and its role in therapeutics revisited. Okay, so we come to the basic question: what is medicinal chemistry? Now, according to Wikipedia defines medicinal chemistry as chemistry of drugs, which is intersection of synthetic chemistry, pharmacology, biological specialties, design, chemical synthesis, development of pharmaceutical agents, including biostatistics and other things. It also includes medicinal chemistry, the organometallic chemistry as a some part of medicinal chemistry, such as saying lithium-based drugs, or say platinum-based compounds such as cisplatin, gallium, and other drugs. So this is basically medicinal chemistry has 2 interfaces. One is the chemical interface and the other one is the biological interface. The chemical interface basically uses chemical biology, enzymology, and then computational chemistry, and then chemistry of natural products, and basically chemistry of small molecules. Okay, and then the biological interface. If you talk about the biological interface, then biological interface includes say computational aspects along with the biological areas such as biochemistry, molecular biology, pharmacology, toxicology, veterinary and human medicine, project management, statistics, and basically at the biological interface of medicinal chemistry, it is an intersection of all these fields are applied to produce one single field, which is called as medicinal chemistry. Okay. So pharmaceutical chemistry basically. So pharmaceutical chemistry. So pharmaceutical chemistries include organic chemistries, pharmaceutical chemistry, inorganic chemistry. So this is the definition. Yeah, there is an interference there. Please close your mic, Miss Priya Jaiswal. So physical chemistries so pharmaceutical chemistries include organic chemistry, pharmaceutical chemistry, inorganic pharmaceutical chemistry, pharmaceutical and organic chemistry. So applied to medicines is medicinal chemistry, or else it is simply pharmaceutical organic chemistry. So pharmaceutical organic chemistry applied is to medicines is medicinal chemistry, or else it is pharmaceutical chemistry, organic chemistry. Other application is organic chemistry of preservatives, additives, excipients, which is not medicinal chemistry but included as pharmaceutical organic chemistry. Then pharmaceutical inorganic chemistry, this is applied to pharmaceutical chemistry to medicines, is included in medicinal chemistry on broad term. Then organic chemistry, then 95% of the molecules are organic in nature, and organic chemistry applied to pharmaceuticals is pharmaceutical organic chemistry, and applied to medicines is medicinal chemistry, as stated above. And then we define medicinal chemistry as intersection of pharmacology, pathology, toxicology, biological sciences, chemical sciences applied to medicines in human, including basically it includes both human and veterinary medicine, and it is called as medicinal chemistry. Okay. So we come on to the next section, which is called as a receptor. So receptor is a concept in medicinal chemistry wherein the drug goes and binds to the special type of organ or tissue in the body, which is called as a receptor. Or every organ and tissue has got a receptor, or every receptive substance is called as a receptor to which the drug binds and produces the biological response. So types of receptor includes ion channel receptors such as ligand gated ion channels, voltage gated ion channels, and then G protein coupled receptors such as metabotropic receptors, and then enzyme linked receptors such as intrinsic enzyme receptors and JAK-STAT kinase receptors. And then receptors are in regulating the gene expression or the intracellular or cytoplasmic or the nuclear receptors. So intracellular receptors, these are all ATP dependent, and basically they give a feedback to the ATP mechanism, and ATP is phosphorylated, and once the ATP is phosphorylated, it produces the response, and then it produces the response to further give the negative feedback, and further to either to modulate the activity of the receptor or to terminate the activity of the receptor. So this is the role of intracellular receptor. Extracellular receptor, it has got amino terminus, lies on the extracellular region. The loop binding sites are situated on the extracellular surface. Smaller ligands bind deep within the extracellular groups, whereas larger bind superficially. Okay. The structure heterotrimeric receptor has got 3 subunits, that is the alpha, beta, and gamma subunits. Then GPCRs, they have 7 alpha helix, and then 3 and the other beta and gamma subunits. Then phosphorylation of channel protein on one or more protein of its subunits can confer both activation and inactivation depending upon the channel and nature of the phosphorylation. So there is a term of phosphorylation. The receptors are phosphorylated and they become activated and basically subsequently again phosphorylated to become inactivated, and this process goes on till the biological response is produced. So you can see here where these are the transmembrane domains. TMD is a transmembrane domain. T for trans, M for membrane, and D for domain. And these are the TMD 1, 2, 3, 4, 5, 6, 7. And I1, I2, I3, you can see here, these are the intracellular loops called as I1, I2, I3, I4, I5, I6, and these are, and so on. So the receptor activation is basically a conformational change in the G protein coupled receptor. This releases the alpha subunit and the GDP replaces GTP. GDP is guanosine diphosphate and GDP is guanosine triphosphate at the alpha and the beta subunit and binds to the particular target and causes activation and inactivation. The receptors have subunit, and this is the this subunit property, or they bind to the Janus kinase enzyme or activation, and these are called as enzyme-linked receptors and so on. Basically, if you are a new learner in medicinal chemistry, so you need to know the concept of the receptor, types of the receptor, and what activity do they play in bringing about the biological response, rather than learning what the receptors are. Basically, what types of receptors are is more important. Okay, neurotransmitters are for all types of receptor. Different types of neurotransmitters show different action. So the way you have a receptor, the receptive substance, receptor, which is the receptive substance in organ or tissue, then on the other side you have a neurotransmitter also, which is a chemical substance which goes and binds to the receptor. So acetylcholine, basically deficiency of acetylcholine is seen in Parkinson's and excess is seen in myasthenia gravis. Adrenergic receptors, these are seen in hyperkalemia and Raynaud's syndrome. Then GABA receptor, this is seen in nystagmus, retinitis and seizure and anxiety. Excess is seen, dopamine is seen in Parkinsonism, and psychosis is seen in excess in psychosis. Histamine, it is a deficiency. There is no deficiency of as such histamine. Sometimes it excess is seen in Meniere's disease, or which is called as vestibular disease. And then you have serotonin, or the aging brain anxiety, and excess is seen in depression and fibromyalgia. So these are various types of disorders, and this is the various types of neurotransmitters where they bind to the receptor and bring about particular activity, and seen in diseases and various types of other kinds of other kinds of disease ailments. Now there is something called as structure activity relationships. Now structure activity relationships are sometimes important. Basically, structure activity relationship means structure and its relation to the biological activity. What role does a structure or organic structure plays in biological activity, and what biological activity does it brings about? So this is basically structure activity relationship role. And here you can see that chemical groups responsible for evoking a target or a biological effect. These determine the biological response they produce in a living body, whether it's a human or whether it's an animal. Then it approaches the nature and position of substituent that affects the biodegradability of several chemical classes. Now, structure activity relationships also determine how the drug will degrade in the body, how the drug will bypass, or whether it will undergo the hepatic metabolism or not. Then for drug discovery and development, it is responsible. Then in case of drug design, for identification of side chain. Then in case of therapeutics, then potency to make the choice of drug treatment. Now, sometimes studying the drug structure activity relationship, you decide which drug to be given to the patients, and this is most being practiced by the pharmacists. Now, this is how the structure activity relationship looks like. You can see this is the chemical structure of acetylcholine. Acetylcholine is a neurotransmitter. You can see here acyloxy group, ethylene group, and a quaternary ammonium group. And you can see the nitrogen is essential and should bear a positive charge in the quaternary ammonium salt to bring about pharmacological activity. And then the size of the alkyl group substituted on the nitrogen atom should be size of a methyl group. Then oxygen atom should be ester-like oxygen capable of participating in hydrogen bonding, and there should be carbon units. There should be 2 carbon units between oxygen. Excuse me. This way between the oxygen atom and nitrogen atom. Now you can see here between the nitrogen atom and oxygen atom there are 2 carbon units. So acetylcholine is a chiral molecule. However, synthetic derivatives of acetylcholine possess chirality. Now, these are various derivatives prepared from structure activity relationship of acetylcholine, and you can see here carbachol greater than buthanachol greater than methacholine, and their biological response is seen in a tissue. You can try it on a frog tissue, or you can try it on a rectocilimus of frog, or you can try it on ileum of rats, and then this kind of what response they produce, the order of potency, how much potent it is given by carbachol greater than buthanachol greater than methacholine. These are the 3 drugs which are produced from the structural variations in acetylcholine by studying structure activity relationship. So adrenergic agonists, these are again class of agents. You can see here there is a metaraminol, there is a methoxamine, and that is phenylephrine. Now most of the time you have cough and cold, and at that time phenylephrine is used as a inhalant class of drug, and by using phenylephrine you can get relieved from your decongestion congestion, and it shows basically a decongestant kind of action. So this is also one of the example. Then the derivatives which are prepared from it are albuterol, pirbuterol, levoalbuterol, and they have various kinds of what we call it as specialties. For example, pirbuterol is an interesting analog with benzene ring replaced by pyridine ring, and they are available as oral dosage forms. Albuterol is RNS mixture, which is called as an enantiomeric mixture, and here the and inflammatory exacerbating airway reactivity to various kinds of spasmogens, and thus shows bronchoconstrictor activity. And then you can see here are levoalbuterol, and it is metabolized by sulfation, rapidly metabolized than S enantiomer, and thus the S enantiomer should be separated from R enantiomer to show bronchodilator activity. What I mean to say is here these are all organic compounds, and this kind of chemistry is applied to medicines to give you what we call it as medicinal chemistry background. Okay, these are other examples, and these are all examples. Now you can see here structure activity relationship of benzothiazide. You can see that is the numbering system is also given here. It is an heterocycle, all of you know. This is an heterocyclic system, and then the second amino group or NH group here is acidic in nature due to the presence of thiodiazoxide at the linkage. C7 group is essential. 2 nitrogen group is essential. C6 is an electron withdrawing position here. You can see here C6 position. Then C3 substitution is a lipophilic position, which you can be substituted by haloalkyl, aryl alkyl, thioether, and other kind of molecules. So the conclusion is, excuse me. So in the conclusion is that the chlorothiazide, hydrochlorothiazide, polythiazide, hydrofluorothiazide, their potencies are shown here. That is, it is 0.8, 1.4, 2.0, 1.3, and their bioavailability is shown. This is another term where you can see that the drugs are usually metabolized, and how much after metabolism they are available to the body. That is the bioavailability concept, and this totally depends upon the chemical structure of the drug. And what you have concluded till now is, we have seen organic structures, you have seen the definition of medicinal chemistry, you have seen structure activity relationship, and basically you have seen how the structure activity relationship has correlation with the biological activity in humans and in medicine use. Now, another example is insulin. Now, this is an enzyme, and you have various kinds of insulin, such as chainase obtained from beef, then pork, and then human. So, this is the various kinds of their structures, wherein you can see that the threonine, isoleucine, and alanine, and at position 30 in humans, it is threonine in place of alanine, and various kinds of replacement you can see here. What arrangements are there in basic there? What is the similarity in between various kinds of insulin obtained from different animals? Okay, now this is the structure activity relationship of insulin, and you can see that B29, 38, the B3 amino acid positions are important, and then C terminus is important, and then B chain B is important. Now this is part of enzymology, and this suits the definition of medicinal chemistry, that it has part of biochemistry, it has a part of pharmacology. What we see that how the structure activity relationship affects the biological activity is a part of pharmacology. Then how the diseases are being treated is parts of pathology. Then how the drugs will show their action is parts of pharmacology, clinical pharmacology, and toxicology. So this is all it is about medicinal chemistry. Then miscellaneous mechanism of action, you can see anti-anginal drugs and other drugs. Now drug interactions. Now these are what you have seen, all kind of interactions in your physical chemistry or organic chemistry. There is covalent bonding, there is ionic bonding, there is hydrogen bonding, there is ion dipole bonding, there is van der Waals hydrophobic bondings, and other kinds of bondings. So all these bondings have various strengths, and these strengths are here, bond strengths are given with the receptor. So covalent bond is 40 to 104. Then basically organophosphates and irreversible binding drugs show covalent bonding, and once they bind, they are poisons. Covalent bonding is mainly shown by poisons. Ionic bonding is shown by mostly reversible and other reversible binding drugs, and all the other bindings are also shown by reversible binding drugs, but covalent bonding is specific. Once the drug is bound covalently, it becomes a poison. So this is what you have neurotransmitters, their structures you can see in Google and other things, and their distribution in body. For example, amines are located in CNS. They have excitatory and inhibitory activity. Acetylcholine, just now we saw the structure. Noradrenaline, we just now saw the structure. Their regions are hypothalamus, limbic system, and reticular formation in brain, and both excitation and inhibition effect. Then dopamine is in basal ganglionic membrane, and so on. Now, concept of prodrug. Now, this is another concept in medicinal chemistry, and it has got vast scope of research. And now, prodrugs have pharmacokinetics. Pharmacokinetic means how the drug will behave with how the body will behave with the drug, and how the parameters are governing in metabolism of the drug. So everything is metabolized in the body, and sometimes we have to what you call it as manipulate this metabolism procedures to produce prodrugs, and this research area has got a vast scope. And so the pharmacokinetic reasons why the drugs are being converted into prodrugs. that is improvement of taste and odor, improvement of physical form, reduction of GI irritation, reduction of pain at site of injection, then enhancement of drug solubility and dissolution rate, chemical stability of the drug. And then you have pharmaceutical reasons which are very specific, such as bioavailability, then pre-systemic metabolism, duration of action, minimal toxicity, and site-specific drug delivery. Basically, prodrugs are the agents which are converted into metabolites, and these metabolites are further active in body. So a drug before a drug is called as prodrug. Okay. So this is how the products are produced. That is the transfer moiety and then active drug, and then transport moiety plus drug metabolism is being carried out, and the drug is being separated. So this is becampacillin. You can see the structure here of becampacillin, which is an organic chemical structure, and it is metabolized in vivo to ampicillin plus CO2 plus this is what acetic acid, I guess. Acetaldehyde. Plus ethanol. So this is active drug. Chemical modification may or may not be active and metabolized to active drug. Now you can see this is another drug which is an anti-inflammatory drug. You can see the chemical structure here. It is Sulindac, and you can see the sulfone group which is being metabolized in vivo to a sulfide bond. Now estradiols, and these are all steroids, and you can see here that 17 alpha estradiol being metabolized to alpha 17 alpha estradiol and so on. So these are the other examples, and then we have stereochemistry. Yeah, the stereochemistry is important. The way we have stereochemistry in organic chemistry, similarly there is stereochemistry in medicinal chemistry also. And in medicinal chemistry, the stereochemistry, there are various definitions such as enantiomers, diastereomers, racemic mixtures. And you can see here, this is a drug chloramphenicol, and the tragedy occurred due to the configuration of conformation of chloramphenicol, because of which the several teratogenic effect was seen in population. That is an old story of chloramphenicol. Then norepinephrine R configuration. Then you have Isan-Stedman hypothesis, wherein the stereochemistry plays an important part, and this Stedman hypothesis states that every drug chemical structure binds to the receptor, and the receptor has got particular site, and unless and until there are fitting areas in drug and the receptor site which are matched, there is no interaction of the drug. You can see here the stereochemistry of R-epinephrine here and the stereochemistry of S-epinephrine. You can see here in R-epinephrine and N-methyl dopa, there are no interactions. The receptor is stagnant. Reasons, sometimes the E isomer is active and sometimes the Z isomer is active. It depends upon the drug. Now, basic pathways for metabolism of the drug, you can see here a dose being administered, then therapeutic site, then free drug, protein bound, tissue reservoirs, unwanted site, and excretion and biotransformation. Now, criteria factors for selection of the drug include pharmacokinetic factors. These are all areas of research. This is not what I'm delivering this lecture, not because these are the basics of medicinal chemistry, but these are the research areas also of medicinal chemistry where you can work in, and you can go ahead with your research areas, and then you can see where what is the scope of research in particular those areas. Absorption, distribution, metabolism, half-life, dose, action of drugs, and adverse drug reactions, that is ADRs. The what we call it as pharmacogenetic pathways, you can see here digoxin, which is GI absorption, T half-life. T half-life is the amount of the drug required to metabolize to half of its volume is called as T half-life. So protein binding 20 to 30%, 90 to 95%, enterohepatic cycling 5% to 25% of the drug, and excretion basically by kidneys and liver metabolism, plasma levels 0.5 to 2.5 milligram per ml, and digitoxin 20 to 35 mg per ml. Now, what I mean to say here is this is a cardiac drug and it has got a steroidal structure, but both of these drugs, digoxin and digitoxin, vary by simply by changing the chemical groups, and their biological activity varies by a great difference after changing the chemical groups. Okay, this is all diazepam. Then fast acting insulin and slow acting insulin, they also vary by changing the physical chemical groups in their structure. Fast acting insulin is called as crystalline insulin, semilantin, then intermediate acting insulin, isophane, insulin zinc suspension, then protamine, zinc, and so on. So comparison of a drug basically here by local irritation. So this is basically a main course of medicinal chemistry. So for beginners, this is not much that recommended. Synergistic effect and additive effect. So you go to a chemist, you buy aspirin plus paracetamol, and that is analgesic and antipyretic activity. You take it and then you get relief from pain. Then antihypertensive effect, some amlodipine and atenolol. These structures I have already shown in the slide, and atenolol and hydrochlorothiazide. These also structures I have shown in the slide. Then some anti-diabetic drugs used for diabetics, that is glibenclamide and metformin and pioglitazone, metformin and rosiglitazone. And then supra-additive effect, that is supramethoxazole and trimethoprim, sulfamidazine and trimethoprim. These cause sequential blockage in case of malaria and bacterial DHFR and so on. And then enalapril plus hydrochlorothiazide, ramipril plus hydrochlorothiazide. These are tackling two contributory factors. Then you have choices of drugs, how to make the choice of the drug. Suppose it is anti-emetic, there is a ondansetron, and safety wise, ondansetron is preferred rather than promethazine, cyclizine, doxylamine, dicyclomine, antibacterial chlorotrimazole, and rather than penicillin G and ampicillin, because most of the patients are sensitive to penicillin. Then anti-malarial mefloquinone artesunate rather than chloroquine and proguanil, because they have chloroquine and proguanil has got more side effects and CNS toxicity rather than mefloquine and artesunate. Then antiviral terbinafine, then anti-anemic drug, that is iron salt, oral salts, and then antidepressants. This is how you make the choice of the drug in medicinal chemistry. So drug discovery, now drug discovery is a part of medicinal chemistry and it has got several aspects. This is basic area of research of drug discovery for medicinal chemistry. Random screening is what you synthesize and what you put in lab, and then you go for biological evaluation, whether you go for antimicrobial evaluation or whether you go for evaluation in rats. That is random screening. Non-random screening include you go for a test, you go for QSAR, qualitative structure activity relationship, you go for computer-aided drug design, and that becomes non-random screening. And then drug metabolism studies, and based upon this I told you in pro-drug approach, you have to go for drug metabolism studies and design a drug. So this is the clinical observation, and from this Viagra was developed, that is for that was for anti-anginal activity basically, but it had some other use, and so it was being developed from a drug which lowers the blood glucose sugar. Now ring reverse. Now you can see here trans diethylsbesterol is converted to 17 beta estradiol, and these are all ring bioisosteric approaches. These are all ring bioisosteric approaches to be studied. Then you have computer-aided drug design and analog design. Now this is separate section. If time permits us, we will deal with it at the end. This part is ended. So thank you for hearing this part, and then we will go to the next part, which is medicinal chemistry as applied science. Now, do you have any questions in first part? Yes, dear students. Do you have any questions? You can type in chat box also, no issues.
If someone have any doubts, so put your question in a chat box. Okay, later on we will discuss with sir. Sir, you can proceed with next topic.
Okay, okay. So medicinal chemistry as an applied science. Now what you right now saw was a definition of medicinal chemistry, and it is an applied science basically. And now let us see its intersection. Now this is organic natural product phytochemistry. Now when this is applied to the medicine approach, this culminates into medicinal chemistry. Now during Corona period, there were many of scientists who claimed that some of the natural products are important and they show immunomodulatory activity against Corona. And that is good, because there should be some drugs also to show immunomodulatory activity. And this is what we had, a licorice. Licorice is a drug, and this is glycyrrhizic acid, which is the active chemical constituent in it. And this showed antiviral activity against Corona. Now organic chemistry, now recovery, now you can see here, this is paracetamol organic structure, and these are paracetamol tablets. So basically it is organic chemistry, no? But it is applied to medicine, so we call it as medicinal chemistry. Now you can see here various kind of bioorganic chemistry. Bioorganic chemistry, you have metallic compounds. Here you have cisplatin. Here you have KP1019, which is a ruthenium complex, and then you have sugar complex here, and then you have bismuth complex there. These are all complexes, and you can see here that this is an applied science. These are all X-ray crystallographic structures of these complexes, and bioorganic chemistry is also a part of medicinal chemistry. Now biochemistry. Now biochemistry is a part of medicinal chemistry, and both clinical and non-clinical, and it is an integrating science. Even the FOA's textbook of medicinal chemistry, which we read, which pharmacy students read, or it can be read by anyone. There is no limitation to knowledge. So you can science or organic chemistry students can also read it, FOA, and this is a biochemical approach book for medicinal chemistry. Okay, now you can see here enzymes, and this is structure of insulin here. Insulin is a part of enzymology. Insulin is a part of biochemistry, and you can see here insulin injection. So this is part of medicinal chemistry. Now synthetic organic chemistry, yeah, obviously not limited to paracetamol. Now you must be having in your lab synthesis of paracetamol only, or synthesis of aspirin only in your BSc syllabus, chemistry syllabus. Synthetic syllabus, chemistry syllabus. So that is not only synthesis of paracetamol, but synthesis of other drugs also. So synthetic organic chemistry is also a part of medicinal chemistry. Of course, then human medicine. Now medicinal chemistry is a part of, I told you, this is, I'm repeating third and fourth time, that dosing, pathophysiology, clinical pharmacology, biochemical approach, pharmacology, actual medicine concepts at molecular level is medicinal chemistry in humans, for humans. Veterinary medicine, yeah, obviously veterinary medicine is also a part of medicinal chemistry. Now you can see the dose convergence here. Now the human dose is given by 37, whereas mouse, hamster, forget about mice and hamster, we have to deal with the pet ones which we keep at home, which are cat, dogs, or sometimes monkeys also, and sometimes horse also. So these are dose convergence from human to animals, and how these dose convergence are given in medicinal chemistry, and it is a part of veterinary medicine and human medicine. chemical biology. Now, the chemical biology itself includes bioorganic chemistry, biophysical chemistry, biochemistry, bioinorganic chemistry, bioinorganic chemistry, and chemical biology is also a part of medicinal chemistry. Now, role of medicinal, this part is ended, medicinal chemistry as an applied science, and this is the third part which we have, that is the role of medicinal chemistry revisited, that is natural products and phytochemical approach. After this, we have computer-aided drug design, and I don't think we will have time for computer-aided drug design, but I will try to cover it if time permits me. Okay. Okay, natural products, NPs, these are called as NPs, as resources in drug discovery and recent advances. These are medicinal and synthetic approach to natural products, and this is also a part of medicinal chemistry. So the history, now here what the data is collected here is from January 1981 till 30th September 2019, and then disease states, plethora of diseases are identified, and from 1946 till September 2019, that is around 73 years data is presented here. Now 75 molecules, out of which 53% were natural products. Then 1981 to 1920, 185 unadulterated natural products were identified. Out of 185, that is 62 molecules were natural products, out of which 35% were natural products, and 58 are synthetic drugs and synthetic drugs, and their ratio was 64.9%. That is a natural ratio percentage was 64.9%. FDA approval status. So FDA approval status is counted basis on biosimilars, whether these natural products are approved as drugs or not. That was given by FDA. And natural products or NPs, their synthetic derivatives still have space. And this is also a part of medicinal chemistry. All kind of what we call it as in pharmacy language, we call it as pharmacognosy, or we call it as herbal medicine, or what we call it as phytochemistry. And same things are called as in basic science syllabus courses also. Okay, so de novo combinatorial chemistry was one of the greatest achievements in medicinal chemistry, where sorafenib and vemurafenib kind of drugs were approved in 2012. Then anti-infective area still remains challenging for natural drug discovery, because natural product drug discovery has not yet identified what we call it as anti-infectives. Then natural product drug discovery, Lipinski rule of 5 is there in the medicinal chemistry. Lipinski rule of 5 approach is there. So according to Lipinski rule of 5, there should be the first 4 rule are not obeyed itself by the natural products. And Lipinski rule of 5 says that 5 hydrogen bond donor, 5 hydrogen bond, 5 or a multiple of 5 hydrogen bond donor, 5 or a multiple of 5 hydrogen bond acceptor, molecular weight should not be greater than 500, and it should obey what we call it as atomic weight should not be greater than 500, and so on. So these 3 considerations are basic consideration of Lipinski rule of 5, and the first 4 considerations are not considered by are violations of Lipinski rule of 5 by the natural product drug discovery or natural products. So this is the Lipinski rule of 5 violations. Out of the 12 FDA approved drugs, that is 6 were of HCV treatment, that is hepatitis C virus treatment. 4 were anti-tumor drugs. 1 was nausea and vomiting. 1 was for cardiovascular drugs. So molecular weight of this compound was from 531 to 894. C log P. Now this is calculated log P value, which is partition coefficient. Log P is log of partition coefficient between octanol and water. Okay, the drug is partitioned between octanol and water, and the ratio is the C calculated log P value. Okay, so in 2014, the data was published of bioactive which violate the Lipinski rule of 5. Now libraries, there are 5,000 libraries of these molecules. Databases, there is Wildman database, there is Paint database. These databases are on Google. You can search this. IMP's database. These are all databases available for natural products, and you can search them on Google for natural product drug discovery or phytochemical drug discovery. The major category here I am representing is B means biological, N means natural product, NB means natural botanical product, ND means natural product which is a semi-synthetic modification, S means totally synthetic, S asterisk means synthetic but pharmacophore from natural products, V for vaccines, and NM for natural product mimetics. So rationale for the subcategory S by NM and S by NM, that means synthetic products with the natural product modification category from natural origin. So here in past 40 years, you can see here the contribution of natural products and medicinal chemistry and synthetic modification of natural products in medicinal chemistry for the drug discovery. Remember that drug discovery is the easiest research in pharmacy or basic sciences. Now, every research in chemistry, if it culminates to drug discovery, then it is wonderful research and most easiest research, although very costly research, but it is most easiest, safest, and the most successful and rewarding research. Now n is equal to 1394, and you can see here that there are natural products and there are several other natural products of origins as shown. Pharmacophore, that is derived from natural products led development to the armamentarium of physicians, therapeutic armamentarium currently developed. So past 40 years, in 2004 there were 24 NCEs. In 2005, there was 52 NCEs. NCEs are new chemical entities. 10-11, there are 33 to 34 NCEs. 2012, 61 NCEs. 2013, 47, 68, 69, 52, 70, 75, and 48 in 2019. How they have contributed, where most of them were of synthetic origin and synthetic natural product modification origin. Anti-parasitic drugs, you can see here that most of them were of natural product origin, and 2% were of synthetic origin, and 6% of synthetic, mainly basically synthetic origin, whereas 2% of from the biological origin, and so on. So this data, anti-cancer drugs, N equals to 321, where you can see here synthetic natural product modification. I am only concentrating on one, were 38%. Anti-diabetic drug in past 40 years, then you can see here this is of around synthetic modification, and then natural product modification, and then synthetic natural product modification, and purely synthetic origin. This is all data. All multiple agents, anti-glaucoma drugs, 19%. You can see here N is equal to 19 drugs were discovered, and then out of which around 2% were of synthetic and natural origin. So we come on to the last chapter. I will have a break for 2 minutes. I want to have some water, and then we will start with computer-aided drug design. So computer-aided drug design, this is the last part, and obviously everyone will be relieved because we are in the last part of this session. I have seen participants also moving out because it is a continuous talk rather than having a interactive session. But sometimes you have to, since we have a professional barrier, basically you belong to science and we belong to pharmacy. So when this barrier is crossed, we can have an interactive session also. And basically you can go, but the advantage of being in science is you can go for any research, whether it's an pharmaceutical research or whether you can it's an any research, but a pharmacy candidate cannot go for basic research, okay, due to his professional barrier. Although he does, he will not be that much successful. So science people have got that much advantage over other professional graduates. Okay, computer used in computer aided drug design. So this is a course also on Udemy. You can go with this course on Udemy for computer aided drug design. This is run by ITOS Foundation, and this is part of it. Introductory part I have taken from that online series. The course is of around 3,000 rupees, but you can here I'm putting it for free to you. And so computers in medicinal chemistry, that is what is used in medicinal chemistry, what is their basic use? So basic use is literature searching such as Medline and SciFinder, structure searching, protein database bank. These are all free. You can search on Google. So you can search on Google, and these you can get on all on Google. Google it out, the names Medline, SciFinder, and you will get the data. Okay, protein data bank, all the proteins are crystallographic structures of the proteins are deposited in protein data bank. SciFinder, all the bibliographic and biographic information is in SciFinder. Sometimes SciFinder has got also utility for what we call it as searching chemical structures also. Okay. So cataloging structure activity data, structure activity, what it is, I have already described you. And so this is applied science of structure activity or application of structure activity relationship to computers and going for computer aided drug design, modeling existing lead compounds, developing new lead compounds. Okay, so is the target structure known? Target structure means target from disease. If it is known, yes, you go for structure based drug design. Otherwise, if it is known, not known, you go for pharmacophore based drug design. Pharmacophore based drug design is also called as ligand based approach. So ligand based approach basically has QSAR, 3D QSAR and qualitative structure activity relationship. Whereas other generate new leads includes proposing new leads and then going for protein structure identification and then going for all what we call it as building QSAR model, docking and other things. And finally going for synthesis of the best fit compound, testing the biological activity and developing the drug candidate. So this is the drug discovery process today. So which has got physiological hypothesis, molecular biological hypothesis, genomics, and then chemical hypothesis. And then you come on to a hypothesis, what we call it as a biochemical hypothesis. And you develop a combination of molecules are screened. And finally, you develop an initial hit, which is the drug or drug template. Basically, what you do in academic research or what you do in laboratory research is finding a template or an hit. It is never finding a drug. Drug discovery takes around 15 to 20 years to complete, or at least 5 to 10 years if it is rational based drug design. Initial hit, secondary mechanism of action, this goes to hit to lead and then first in vivo, and then you can go for a trial, that is finding out a lead or finding out a hit or finding out a template. Okay, lead optimization. Now this is another part in drug discovery. Once the hit or lead or the template is identified, you go for lead optimization, and lead optimization is done for potency, selectivity, physical properties, pharmacokinetic properties, metabolism, oral bioavailability, and then synthetic ease, and then scalability. Scalability is technology transfer phase, and basically you should have drugs, and you should have drugs which are scalable, which are can be produced on large scale. And scalability is also for medicinal approach, also scalability in terms of diseases also. Okay, so pharmacology in multiple vivo models, chronic dosing, and then preliminary toxicology, and then best development of drug candidate and backups. So this is how the drug discovery process works. That is target identification, hit discovery, and then hit to candidate optimization, then preclinical study, and then clinical study, and then the drug is put into the market. Of course, with approvals from FDA and state FDA, national FDA, US FDA, and other FDAs. Now this is RDD, this is a rational drug design, and computational chemistry approach is the part of a rational drug design. And you have 3D crystallographical structures of proteins deposited, X-ray crystallographical structure of proteins deposited in protein database bank, and from there you can get the structure, load it on the computer software, and go for the co-crystallized ligand structure, and then you can go for docking and other kinds of structure rational based drug design. So rational based drug design includes two types, that is structure based drug design or structure based ligand design, wherein the macromolecule of the drug is being utilized, or macromolecule of the protein is utilized, or macromolecule of the receptor is utilized, and then the docking studies are carried out. So computer-aided drug design basically is identification of lead compound, optimization of lead compound, evaluation of delivery and metabolic issues, optimization of synthesis, and animal and clinical testing. So computer-aided drug design computational approach has got three kinds of computational methods: QSAR, confirmational analysis, pharmacophore studies, database searching, and structure-based drug design. And then combination of techniques useful to compared to any isolated technique. Okay, computer-aided drug design. First, computer-aided drug design. First step is computational search, confirmational search, computational search, and searching all kinds of softwares, and then you have pharmacophore preceded 3D QSAR studies and database searching. Then modeling existing lead compound includes QSAR and then pharmacophore model development. Then you have de novo drug design, which includes starting from the scrap. Starting from the scrap is de novo drug design, and it includes database searching, combinatorial library design. And you have structure-based drug design, where the molecular docking, calculations, binding energy estimations, and molecular designs are being carried out. So this is how structure-based drug design carried out. You have the crystal structure of the receptor, you go for computational analysis, then ligand-based structure, ligand design, ligand docking, biological testing, candidate, and clinical studies. Traditional drug design includes lead generation and then natural ligand screening, then biological testing. The drug is called as a ligand because it is a drug. Okay, so biological testing, synthesis of new compounds, and if promising, you have preclinical studies. Structure-based drug design, this is explained. Then structure-based drug design too. Then you have a high throughput virtual screening, wherein large libraries of the molecules, organic structures are generated in the library, and then this library is being screened for hit. And the zinc, ZINC, the zinc you can search on the Google, and this is the database which is used for high throughput virtual screening. Sequence analysis, this is the what you have seen up till now is the molecular part of, or the ligand part of, or the drug part of computational chemistry. Then sequence analysis is the protein part of computational chemistry. And here you have what we call it as bioinformatics, and sequencing of genes and sequencing of enzymes, structures or enzymes, enzyme sequences, amino acid residues and others based upon in silico, that is in computers. Homology modeling, these are the modeler and Swiss model. You write this is a free information. This is very important for you if you want to go for drug design. Modeler software is there, Swiss model software is there. These are all free softwares, and you can go this for from Google search, and you can go for homology modeling. Play with the software and you will learn how the software works. secondary structures, alpha and beta helix structure prediction, and so on. Then protein docking is one of the part, protein ligand docking is one of the part. Other uses is stability of this thing, stability of drugs, determination of stability of drugs and other things. For this also computational approaches are used. Similarity searches. Now 95% of the world works on similarity. The day there is a difference, it leads to war. So basically everything works on similarity. If you differ, it's a war. So there are similarity searches in organic structures also. And in similarity structure, there are substructure search, there is electrostatic structure search, there is 2D and 3D substructure search. And this is also a way of optimizing ligand. Then physicochemical modeling, you have Swiss PDB. You can go for check this on Google. And then hydrophobicity and polarity can be searched on Google. And then finally you have drug bioavailability, and there is a PAS server, PASS server, and on the PAS server you can go for the studying bioavailability. So this is my basic qualification. Out of this, I am now currently doing a PhD in medical sciences, and I have already done MD in alternative medicine, BA, MS in alternative medicine, but these are non-regulated degrees by UGC. These don't come under the purview of UGC, but are practicing degrees. And I am already PhD in pharmaceutical sciences also. And with this, thank you, and thank you for your patience and listening very carefully. And please let me know if there are any questions. You can ask for questions. We will go for questions.
Yeah. So anyone have the doubts regarding today's topic? All the participants are freely ask any type of the question to our resource person without any hesitation. Sir, my one question regarding with the drug discovery. Last 3 years back, when the COVID comes, there is a panic one to each and everyone.
Right.
But after immediately the 6 months later on, there are the certain drugs which are get launched in the market. And as per our guidelines, any particularly the new drugs or the API, when it is get launched in the market, it requires near about 15 years, or when you go with the national drug delivery system, so it required near about 10 years. How this is possible during the last 3 years back, whenever the pandemic situation comes?
No, in pandemic situation, actually, basically these drugs were off-label use was coronavirus. They were antivirals. Their basic category was antiviral, but their off-label use was for coronavirus, and they were in pipeline only. They were in pipeline from last 5 to 10 years. But since the pandemic arose, they were launched in market for emergency use by FDA. All the bottles and the labels on the drug was labeled as for emergency use, and they were not as such for routine use. Now you see remdesivir, this is not used now. Remdesivir is out of market now. Remdesivir was used for corona phase, but the moment it showed a toxicity, it was withdrawn. And now the other potent drugs such as rotonavir and then liponavir, these are now being used in market now for corona. And also ostelamir and other which are used for swine flu. So basically they were 10 to 15 years old only, but FDA launched, US FDA launched them for emergency use. They were in pipeline only, and their off-label use was for coronavirus.
And sir, regarding you talk about the computer drug designings are there. So it is an any type of the software software which is easy for the student to targeted the hit candidates one whenever there will be the drug discoveries are there, or you have to go with the certain fees paid research lab
No,
for
there is
their
a,
screening out.
yeah. I understand your question is very valid. There is Autodoc software, AUTODOCK. Okay, and there are various Autodoc software tools are there. There is Autodoc Vina tool, Autodoc Python tools, and other tools. These are free softwares, but only thing is that they are very time consuming. One ligand, batch ligand process takes very long time. Okay, so these are free softwares, you can go for with them. And secondly, they are third party softwares, so you can show in your NAC also that you have enabled some of your systems in computer with these softwares, because they are license based, and the license is from Scripps Research Institute. Okay.
Okay, sir. Yeah, thank you. Anyone participants?
Any more questions?
No.
Hello, sir. I have one question.
Yes, please.
So we are our student belonging to chemistry, and particularly organic chemist will go for pharmaceutical industries.
Okay.
Okay. So please tell how they will get absorbed in a pharmaceutical chemistry as their subject organic chemistry, and how they will get a job in pharmaceutical industries on the basis of their subject, and what are the conditions and what is the procedure they get absorbed in industries as pharmaceutical company as a organic chemistry student particular. So
Okay.
please.
Okay. First thing I will make you clear, there is no relation between your curriculum and what you do in industry. These are two totally different scenarios. Okay. In curriculum, you will learn all synthesis and everything, but what theoretical yield and percentage practical yield you will learn. Okay, preparing theoretical yield and percentage practical yield in synthesis, only that part is important in industry, because in industry, in pharmaceutical chemistry, if you are dealing, so in API industry, API is active pharmaceutical ingredient industry, where there is a synthesis of huge, there are tanks, and there is synthesis of intermediates, and there is synthesis of drugs on bulk, maybe around say 1,000 tons, or say I don't know, 10,000 tons. So the moment you will do BSc chemistry, you will be absorbed in organic synthesis division, and in and day out you will be referring Badger and Banchero textbook, which is an organic engineering chemistry book, pharmaceutical engineering chemistry book, and based upon that you will have to deal with how the process can be modified, synthetic procedure can be modified, how it can be patented, how the synthesis can be improved, and how the theoretical and practical yield can be improved. And this is how the API industry works. So this is one of the aspect. And if you are BSc chemistry, you find place in pharmaceutical industry in formulation and development, because there is scope in drug interactions. There is scope where organic chemists play a major role, because they have a better hand rather than pharmacy graduates in organic chemistry. Okay, did I answer satisfactorily?
Yes, sir. Sir, is there any exam for the for industrial pharmaceutical industry for to get a job there? Is there any entrance exam or interview?
There is interview itself.
Okay.
There is interview, and then what you can do is there are certain organizations which give certifications. There is Wipro and other organizations which give certification in your some areas of pharmacy, and those certifications are considered by industry. So basic science plus a certification in say quality control or quality assurance, or say then chemical synthesis, this will lead to you a good industry. Now I don't want to take the names of these institutions, but I belong to ATOS Foundation. Even if you do ATOS Foundation courses, you will end up in industry, no doubt. These are approved by industry. And there are other organizations also who run these courses, and you can do these courses for a good career in industry.
Okay, thank you, sir. Thank you very much.
Yes, any more questions?
Now at the last phase, top thanks. Now I requested Manisha Madam give the award to
आवाज येतोय का माझा? Am I audible, sir?
yeah. Continue.
Respected sirs and all the participants, it's my privilege to have been asked to propose a vote of thanks on this state-level online webinar on behalf of the Art and Science College, Pulgaon, Department of Chemistry and Department of Microbiology. Extend a very hearty vote of thanks to Dr. Amit G, sir, Professor and Head Siddhant College, Pune, who blessed us with their presence and took valuable time from their busy schedule, where he mentioned the way he explained the topic is exemplary and has no webinar can become more successful with a single person. So I can extend my big thanks to our management, Principal Dr. V.V. Hadge, sir, for constant support. And thank you, student, for making this event successful with your contribution. Thank you, sir.
Yeah, I want to say something. I am very much overwhelmed with your invitation, and thanks a lot to the Art and Science College, Pulgaon. Thank you to the principal, and very much gratitude to Panginwar sir and management of Pulgaon Arts and Science College, Pulgaon, and hope we have a successful endeavors in futures also. With this, I say closure to this webinar.
Okay. Yes, with the permission of resource person, now I end this meeting.
Okay.
Yeah. Thank you, sir. We will keep in touch in future also. I always require your help because you are you are a motivator for us.
Banginwar sir is a very good friend of mine, so we don't keep grudges also, and always help each other. So I thank so special thanks to him also.
Okay, thank you, sir.
Thank you.
Okay.
How to stop this? Stop video.